record on computer. Good. Let's start because I am late for as you know, we always finish earlier. Oof, maybe what is this? Let's see. This thing. Okay, I am going to use. Do I need it? Is it better? Yeah. Okay. I don't like hearing my voice on the microphone. Let's see. Do you still hear? Is it now? No? Now, yes. Okay. So, how I do this? So we are going to finish today microbial eukaryotes, which is very short. So I reduced this lecture as much as I could to facilitate your life because there is a lot of information. Viruses are fun, but this is not. So, I mean, I like viruses. They're cute. There are 7 groups of microbial eukaryotes, and I cherry-picked the most important parts for you to know. If somebody wants to learn more, obviously there is a book that I posted that is on eClass, and over there you have a lot more data, but I thought this is the most important for you to know. So, 7 groups. Diplomonadas, it's from the closest, that was called Diplo. Parabasalids, Euglenozoas, Euglenides, you already heard about Euglena viridis, Alveolates, Stamenopil, Amebozoas and Cercozoas and Radiolarias. Amebozoas you already know about, that's amoeba. And there are 2 types of amoeba that you will know about. So, what are diplomonads? Diplomonads. So the name diplo tells you something. It has 2 nuclei. So this is, now we talk about the eukaryotic cells. So there is nucleus involved. So what they have, what is their characteristic is something like reduced version of mitochondria called mitosomes. And they lost genes for many metabolic pathways. What you need to know is that they have reduced mitochondria, which are mitosomes. Basally, and I don't have any example I want you to know. I mean, there are, but I'm not bothering with that. Parabasalids, there are examples. It's the Homonus vaginalis, and it's a famous parasitic organism, but it's technically an animal. It's a parasite, actually. And what they have, and that's why they're called. So the same how diplomonads are called diplomonads because they have 2 nuclei. These guys have parabasal body, which is there to support the Golgi apparatus. And their characteristics, so the thing called diplomonads don't have mitochondria, they have hydrogenosomes. So again, energy generating organisms. And Trichomonas vaginalis is a famous example. Euglenozoa, you all heard about Euglena viridis, but there are 2 types of euglenozoa, kinetoplastid and euglenids. Euglenids is Euglena viridis. It's a famous organism that has chloroplast and has 2 flagella and eats bacteria. So it feeds on bacteria via phagocytosis. It has very flexible cytoplasm and engulfs bacteria. Kinetoplastids, so they're named by the mass of DNA that is present in mitochondria. So that's why it's called, that's called kinetoplastid. It's a large mass of DNA. Euglenids are the ones that I am focusing on. Euglena viridis is representative. It contains chloroplast. So in earlier times, that was so it was disputing whether this is an animal or plant because it contains chloroplast. But it's considered an animal, so it moves around and it swims very fast. In dark, cells of Euglena will lose the chloroplast, so and then they live as chemoorganotrophs. The way how they feed with bacteria, and that's their characteristic, is that flexible cytoplasmic membrane, and thanks to that flexible cytoplasmic membrane, they can easily engulf bacteria and perform that phagocytosis. Alveolates so the name comes from cytoplasmic sacs that are located just beneath cytoplasmic membrane. It's called alveoli. That's why they're called alveolates, alveolates. So they have that like a literally like a belt beneath the membrane, and that alveoli, like sacs beneath the membrane, are helping in maintaining osmotic balance. So they control water influx, efflux. There are three types of alveolates, and I'm pretty sure you heard about some, especially about apicomplexans, but you just didn't know that they belong to the alveolates. They can be ciliates or dinoflagellates or apicomplexans. So, what are ciliates? Paramecium. That's you probably heard about paramecium. It's considered microbial eukaryote. It's an animal. What are dinoflagellates? Again, they're all alveolates. So they are those are these like algae that are forming so-called red tides in coastal zones. So they have that characteristic algal blooming. So this is how it looks. And apicomplexans is something that you probably heard about. Again, you didn't know they are alveolates. Toxoplasma gondii is the example. So it's a parasitic organism. So and also plasmodium species that are causing malaria. So what they have are structure called sporozoids. And those sporozoids are used for transmission of parasites to an host. So if you ever worked with toxoplasma, you will see these sporozoids in brains, like in forms of cysts. I had a chance to see it. So they form like a cyst in mice brain. Not fun to see. But when you work in immunology, you have to see it. Which is great to be, that's why it's great to be in microbiology. You don't have to see that. Staminopils are also so mostly algae. So diatom, you heard about diatom and algae, Oomycetes and golden and brown algae. And I will just touch base on them briefly. So diatoms are probably the most famous type of staminopils. Unicellular phototrophic eukaryotes. They are major component of planktonic microbial community in marine and fresh waters. Oomycetes are known as water molds, and golden and brown algae are called chrysophytes. So golden algae are called chrysophytes. Brown algae are not microbes. So I'm just mentioning them. I'm not talking about them because they're just not microbes. Cercozoans are you probably saw this and you can see it if you go to the biological sciences. These guys are exposed everywhere, foraminiferous. They are really exclusively marine organisms. They look like shell and they're very nice decoration in the shelf. My son loves them. You can see them on the second floor. Amebozoas, amoebas. Again, everyone heard about amoebas. Probably nobody talks about gymnamoebas and entamoebas specifically. But they are famous. They're like microorganisms, and their characteristic is that they have those pseudopodias, which are like a little seed, and they move around. So entamoebas are more important for us because they are parasites. Gymnamoebas are free-living protists, and they're called protists, like micro animals in aquatic and soil environment. For us, more important are entamoebas because they're parasites. We talked about literally all of that, organelles, protists, and what we have now are fungi and red and green algae. And I am just briefly going to mention red and green algae and fungi, simply because, especially because fungi are really not microbes, except the yeast. So yeast is microbe, but again, there are other classes where you learn more about yeast's physiology, and yeast is significant from biotechnological perspective, which if somebody wants to hear more about, we talk about that in the microbial. So there are many species of fungi described. They are large and diverse, very diverse, so they have very complex physiology. They are different from protists, and they're literally the group that is most close to the animals. You will see them in soil, in dead plant matter, and they play the critical role in mineralization of organic carbon. Many fungi are known as plant pathogens. Many of them are causing diseases in animals and humans. Many fungi are actually microorganisms. Like one of them is Candida. That one can definitely call it our parasite. Many fungi are establishing symbiotic association with plants, and they participate in mineralization, in like plants acquisition from minerals in soil. Many fungi can benefit humans through fermentation and synthesis of antibiotics. So they could be problematic, but they can also be our friend. But they're not, majority of them, like they are so diverse, and many of them are not microorganisms. In terms of green algae and colonial green algae, so the Volvox is the one that I'm mentioning. So even though technically it's not microorganism, but it's interesting from a social perspective, like protist social perspective, because this is so Volvox is a colony. You probably heard about that in other classes. So many, like several hundreds of cells with flagellum. Some are motile, some of them are carrying photosynthesis, others are fertilizing the production, but they look like this. They float like a big shining ball that floats around the ocean. So it's not really micro, but like cells are like if you separate them individually, they would be like microbials. So in summary, what did we learn about microbial eukaryotes, except that they are much better? I'm kidding. So here we have organelles of eukaryotes, chloroplasts and mitochondria, or hydrogenosomes. Now we introduced hydrogenosomes as a we introduced hydrogenosomes in our lectures. Chloroplasts are functioning in photosynthesis, mitochondria, and hydrogenosomes in respiration or fermentation. So we saw, for example, the parabasalites have hydrogenosomes. So all three of these organelles were initially bacteria, and they established residence in the primary cell, which is endosymbiosis. Ribosomal RNA sequences usually not used to design phylogenetic trees of eukaryotes. So not 18SR RNA, you find other genes if you want to design their phylogenetic tree. So it's not same like for prokaryotes. Diplomonads, first of 7 groups that we touched base today, unicellular flagellal protists. Parabasal is like trichomonas. They like introns, so they lost a lot because they're parasites. Euglenozoas, flagellated protists, kinetoplastids, and euglenids. Euglena viridis is the one that I am going to insist on. Alveolate characteristic is the cell beneath the membrane, which helps in the regulation of osmotic balance. You have ciliates, dinoflagellates, and apicomplexans. Again, apicomplexus is plasmodium toxoplasma gondii. Pills, and Oomycetes diatoms and the golden and brown algae. Most important, and the one that I asked last year, is amoebozoas, like amoebas, gymnamoebas, and entamoebas. And the fungi I never really asked, so I'm just giving you FYI, because again, not microbes and very diverse. So that's all from eukaryotes, and now we will move to viruses. But of course, if there are any questions, feel free to ask. Yes, Adam? There might be something that I don't know, like I can speeches at the start of the time, but from my knowledge, it's either hydrogen or one. But good question, actually. More questions before we move to the. Okay, viruses. Who loves viruses? Who likes to have one? Nobody. Yeah, we had recently everything, like all our lives were shaped by one of the viruses. You're aware of that. For you guys, but for me, pandemic was the most depressive time of my life. Like, yep, Brittany is laughing because she remembers me at the time. Yep, 9 a.m. and I'm already in my world, which includes a lot of fermentation products from bacteria. Yeah, I am generally, I can't, yeah. So I'm saying it professionally. I like people, and when there are no people around me, I don't function, which is the reason why I got pregnant over pandemic. I did, yeah. I mean, either people got divorced or got pregnant, right? There is no third option. I got pregnant. Like, what will you do? That's you're focused on each other. Either you fight and divorce or you just make a baby. Uh, anyway, which is great that there is no viruses anymore. We are all happy and healthy, but we also need to know what virus is. Again, there will be many repetitions here because you all heard about what virus is, and again, you all probably know that virus is not alive. Technically, there is no kingdom, there is no membership, so it's very. It's not plant, it's not animal, it's not bacteria, it's not fungi. What is it? It's a virus. So what makes me most angry is when doctor gives you antibiotic when you have viral infection. If you're a microbiologist, you freak out, right? Dude, it's a virus. Like, no, you don't get it. Like antibiotic does not kill virus. It's not bacteria. So, but people just don't think about that. So again, virus has no kingdom, no membership. It is literally infectious agent that contains nucleic acid, can be DNA or RNA. It can be double stranded, single stranded. We'll talk about that. And protein code called capsid. It is obligate intracellular parasite. Technically, it cannot survive outside of the cell. There are some. I read somewhere that COVID was able to survive a few hours out, like on the surface, but virus actually needs living cell to survive because it lost many genes. And hosts can be also bacteria, but can be animals and plants. Bacterial viruses are very specific viruses called bacteriophages, and the viruses are known as the small microbes. So again, a fun fact that you need like 500 million rhinoviruses, which is a common cold, to cover the head of a pin. So they're very small. That's the point. So shape and size are various. So these, if you look them like this, they look very nice, very colorful. They're actually not. They what you need to know is that they have two essential components. Again, genetic material is required, can be DNA, can be RNA. It is RNA virus. And they have protective coat, which is surrounding that nucleic acid. And that's all they have. So it's infectious agent. What are their characteristics? So no nucleus, no organelles, no cytoplasm, no cell membrane. It's not a cell. It's a virus. That's why you can't put it in any kingdom. And the way how they exist is literally hijacking that cellular machinery of another living organism to reproduce. So they take genes from the host to reproduce, and they can inject genetic material into the host. So, and very often that's the point, like they just reintegrate into the host material, but we will mention that when we talk about the period. They can be either enveloped or without envelope. If they have envelope, they are enveloped viruses. If they don't, they are naked viruses. Again, they still have to have protein code and nucleic acid. Some of them have outer envelope, but that envelope can really vary. That envelope can be membrane derived from the host cell. Like when they are leaving the host cell, if they don't break host cell, they just pick up, like they just bud out of the host membrane and they pick up host host structure. Envelope can have specific spikes, which are very unique characteristics of certain viruses, like virus influenza, which is a cold virus, has those H&M spikes. And spikes can help in attachment and make them sensitive to chemical actions of the infectants. So those spikes are usually target of antiviral meds. So in terms of evolution, how the virus came up, it's very weird. Like it's weird to explain because it's not a cell. So how did they, again, the even bigger question for me is how prions emerged, or like that's even that I'm not touching that in this class, but it is a question, $1,000,000. There are three hypotheses how viruses show up. The one is early virus hypothesis. So basically, the hypothesis is that at the beginning, there were replicative elements that were preceding early life forms, like DNA, RNA, and the virus evolved from these early replicative elements. That means like virus was before everything. That's a called the virus early hypothesis. Second one is the regression. So, which means there was degeneration of cell. So the degeneration of cell in the reduction of cell was a base for viral rise. And later on, so it's like a reversive evolution. Later on, they just lost genes and acquired parasitic lifestyle. That's the regression hypothesis. And the third one is escape gene hypothesis, is called early hypothesis, escape gene hypothesis. It's like cellular genes acquired ability for self-replication and spreading. And then they picked up protein code, which literally means like escape, like a gene randomly escaped and became independent of the cell. So whichever is, I don't exactly know which one is correct. The thing is that you can't really know, so it's a debate. And again, that's why I will never ask which one is correct. But what I might ask is to explain all three hypotheses. What is the structure of the virus? I'm never testing you to know the sizes. You need to know it's very small, but you already know that. And the way how you see them is through electronic microscope, like you can't see them with the naked eye. The BIOL is like infection particle of the viruses. It contains nucleocapsid, which means DNA and RNA with the protein capsid, like protein code. Protein code is called capsid, so that's nucleocapsid, that's virion, infection particle of virus. Some viruses have only nucleocapsid, like only nucleic acid and only protein code. Some of them have additional components, like envelope is one of them. If there is no protein code, how we call? I mean, this is not in notes. Does somebody know how we call infection particle that does not have protein code? Only infectious RNA. Does that kind of particle exist? It does. It's called viroid. It's and so you have viroids and you have prions. So viroids are just infectious RNA. Prions are just infectious protein, and that prions are causing mad cow disease. And I don't know much about that, but it's very interesting. It's very interesting from like a physiological perspective. If you like immunology and if you like pathogenic side of microbiology. Again, but it's like in order to be infectious, virus has to have nucleocapsid. Additional is envelope, which is outer membrane flexible layer. If virus is animal virus, that envelope originates from host cell. So during viral exit of host cell, it gets that host membrane envelope. And envelope's characteristics, again, sometimes they get like a viral ID characteristic. So very often they have like a neuraminidase, which is enzymatic activity, and they can play a role in nucleic acid replication. So that envelope that very often can originate from the host, but not always, can usually be used as a viral ID because of those specific characteristics, like specific enzymes like neuraminidase in influenza. And those envelopes are kind of viral decoration, very often used as viral ID. Again, nowadays you do sequencing and you know which virus is you are dealing with. So now it's accepted that a variety of virions have enzymes, like many of them. So in terms of viral enzymes, like many, like one of the characteristic viral enzymes that I am going to usually ask, because I think you already know that, and this is not on the envelope. So it's a reverse transcriptase. Why is this? So does somebody know which virus has reverse transcriptase? Yes? It does, but there is, yes, but there is one that is famous. Yes, yes. HIV is very famous by reverse transcriptase. Like you're right, yes, but also like HIV is famous virus that has reverse transcriptase. So and that's a well, actually here. No, sorry, I didn't see that I actually put it there, but you trick. Okay, I'm kidding. So again, again, to me, talking about viruses is literally like making a piece of puzzle. So you have surface markers, you have receptor sites, and so when virus is attaching to the cell, it's to me that looks like a antigen antibody or puzzle making. Like virus has those spikes, and there are certain receptors, and on there are certain places on host cell, and then they literally match like a puzzle. So virus evolved those spikes that function like a puzzle to recognize specific receptors on host cell, and then they make that teeny tiny interaction so virus can be ingested and then make a mess in our organism. So one of the examples that I always bring is the attack of rabies viruses on specific parts of like a brain cell. Rabies virus is one of the most dangerous viruses that, up to my knowledge, is not curable. It like in the developed country, in countries that are under development, like my home country, if animal gets infected with this virus, which happens very often, like you just have to put animal down, and because then it's like if people get affected, people will die as well. So there are vaccines, if I am aware, but this is very common for dogs that are participating in hunting because they get attacked by fox, and fox are carrying rabies virus. Sizes of viruses are really big. Like obviously, you don't have to memorize all of that. There are different sizes and shapes of viruses. There are some giant viruses that can even be close in size to the E. coli, but there are some very small viruses like rhinovirus or HIV that are very small, like much smaller. Even if something is big and it is size of the bacteria, it's still a virus because it doesn't have cellular structure. It has a virus structure, so it's not cell. It's not bacteria. Viruses can be classified based on morphology. Morphology means like you see them in electronic microscope. They can have helical, icosahedral, and complex symmetry. Complex means weird symmetry, literally. So, but that's how you can classify them. And again, when you look them like this, like they look very nice. Like helical viruses, that's tobacco mosaic virus. Polyhedral viruses, adenovirus. So, like illnesses of pink eye that my child gets very often. Uh, Influenza virus is spherical one. That's the one that we get vaccinated against. Complex virus, bacteriophage. So I'm saying it's a complex symmetry because it's weird. It looks like a little alien, in my opinion. And I personally really like bacteriophages, and they're infecting bacteria, which is obviously why I like them. And I'm going to touch base on them more because they have an interesting life. Again, just a different structures, different symmetries, different shapes. And that's how you can classify them based on that. Or you can classify them based on replication. So, which means based on nucleic acid present. It can be RNA or DNA viruses. Majority of viruses are RNA, including COVID. And inside the capsid, they actually have either single-stranded or double-stranded RNA. So it can be either or. So I think this is the only organ, well, the only structure that has double-stranded RNA. Usually double-stranded is DNA. And the DNA viral genome also can be single and double. Sometimes it can be circular and single-stranded. So again, viruses are very diverse in that area. So usually we say DNA is double-stranded, RNA is single-stranded, but viruses can have single-stranded DNA and double-stranded RNA. So they are quite unique in that structure. General pattern of virus replication cycle can be, so it's way more complicated, but I am saying it in very simple way so you can understand. So the first one will be attachment, obviously, like a puzzle, attachment to the real host cell. And then you will have a viral entry or penetration when viral nucleic acid actually gets into the cell, into the host cell. Next one is synthesis, when viral nucleic acid actually uses host enzymes to make like a copies, and literally it takes advantage of cellular machinery of the host to make its protein coat that will surround nucleic acid. Then you have nucleic acid that replicate, again, it uses host enzymes That's why it's parasite. And then you have a sample of nucleic acid plus proteins that were made in the host cell. And then newly formed virions are going to be released to the cell, which could cause host cell to break, or it just virus just buds and takes out envelope. Again, in order to get virus attachment, you have to have specific receptors. So that varies between naked or enveloped virus. They can be either on envelope or on protein code, again, depending on the virus. But the point is that attachment has to happen into the correct host cell to function as a key lock system or enzyme substrate or antigen antibody. The next step is viral entry or penetration. The way how it can be done is fusion, endocytosis in vesicle, or simple injection of nucleic acid. It is a fusion of a viral envelope with a host membrane. It's like a hydrophobic to hydrophobic, so they literally they fuse, like viral envelope fuse with a host cell, and then all nucleocapsid enters. Endocytosis in vesicle, so basically the endosome inside the host cell help in the viral uncoating. But again, virus has to come into the cell. Endosomes are inside of usually eukaryotic, not usually, it is in the eukaryotic cell, or virus just injects nucleic acid through it's like a needle system. Synthesis, which means using a cellular machinery from the host to reproduce nucleic acid and to make proteins for the code. So virus cannot reproduce without host machinery. It cannot do it by itself because it doesn't have enough genes, so it's a very small genome. So it has to invade host cell and to take over cell activities. Usually that leads to the cell weakness, so usually sometimes even to destruction, like a breaking the cell. So literally virus enters the cell and takes the advantage of cellular machinery, which exhausts itself energetically. And the viral cycle inside the host cell can be lytic and lysogenic. And this is very specific for bacteriophages, which I'm going to insist on knowing. So what means lytic, what means lysogenic? So in shortly, lytic means cell dies. Like virus reproduces, takes the advantage of cell machinery, breaks the cell in death. Lysogenic means viral comes into the cell, integrates into the chromosome, or just persists there. Then something happens, induces viral replication, virus just comes from lysogenic, becomes lytic, and kills the cell. Or it can forever persist as lysogenic. And this is what I'm going to insist on during the exam, lytic versus lysogenic infection, lytic versus lysogenic cycle. So during the lytic is bad. That means the virus enters the host cell, makes a copy of itself, and causes cell to burst and lyse. In lysogenic infection, and again, bacteriophages can be lytic or lysogenic. Very often, lysogenic phage can become lytic under certain induction. So virus enters the cell, but it doesn't lyse it. So it remains dormant. Sometimes it integrates into the genome and just persists there. Sometimes it can just do nothing, just sit there. It's dormant. Usually it gets integrated and then it copies. It still uses cellular machinery, but it does nothing bad. It just sits there. So and genome is there. And you will see if you do analysis of bacterial genomes, there are many integrated, but you will see many bacteriophages genes that are all around the bacterial DNA. They did nothing. They just got integrated there and they became and we call that like a viral islands inside of bacterial genes. And it's very nice because you can actually employ that in a certain microbial treatment. If it's stable, if it's stable, nothing will happen. But if there is some stressful condition from lysogenic, it can become lytic, which means cell can literally die. Again, this is something that I will probably, that's what I asked last year. In my time, we were taught that viruses cannot survive out of the host cell. As the science was progressing, we learned that some viruses surprisingly actually can survive on the surface outside of the cell. It will depend on the moisture and the temperature, and obviously on the surface. They tend to live longer on the water-resistant surfaces, like a stainless steel and plastic. It's very weird. Cold virus, surprisingly, this was weird for me too, can survive for several days indoors, uh, but its ability to cause infection drops significantly. Viruses can survive for several hours at low temperature. Enteric virus apparently can survive a few weeks. Again, learned in past that that's not possible, but there are scientific studies that show that actually, surprisingly, it can survive quite long, especially enteric viruses. And this is that picture that I really like when Batman slaps Robin. It's antibiotic. Yeah, I have a virus, take antibiotic. It's a virus. So viruses disrupt the body's normal balance. They're usually causing a problem in our organisms and bacteriophages in bacterial organisms. But you do not treat it with antibiotics because it's not bacteria. Antibiotics are antibacterial agents. So, what are positive aspects of viruses? Again, as usual, we found a way to exploit viral features. So, some harmless viruses can carry good genes into the cell. And if you think about CRISPR-Cas system, it stands for bacteriophages that were found into the bacterial islands. Also, viral vectors are used in genetic engineering, like when you want to do genetic modifications of eukaryotic cells, like for example, in PCAR cell therapy, which is anti-cancer therapy, very famous. You need to have viral genes because they have that ability to infect the cells. So phage therapy is, and the bacteriophages are used in production of recombinant proteins very often. So a bacteriophage along with antibiotics to help with antibiotic resistance bacteria. Some don't really have to memorize all of that. Please don't think that I'm asking you to know all of these viruses. They're just examples of human viruses, and obviously COVID-19 is the first one. And I made the lecture last year, and I was still on the PTSD of COVID. HIV, meningitis, pneumonia, all those viruses, many of us got familiar with. Me as a mother of a toddler, I know all of them. They're my regular tenants. Except HIV, I had everything else. Not chickenpox. I had chickenpox, but he didn't. Yeah. Yeah. Did you all have chickenpox, right? Yeah. I had mine when I was 24. Yeah, it sucks. I got it 2 days after, actually 1 day after wedding. Yeah, it was bad. I hated my husband 24 hours after wedding because I was scratching myself and he didn't let me. So it's not fun to get chickenpox when you are 24 years old. Animal viruses, bird flu, avian flu, so many different viruses. There are a lot of different strains. Some of them infect humans, many of them don't. Plant viruses, tobacco mosaic virus, it causes mottled browning of tobacco leaf. PMD virus has significant promoter that is used in genetic engineering because it's very strong, but it can infect other crops as well, especially tomatoes, but obviously tobacco is their main goal. Just a brief table, what is the difference between bacteria and viruses? So you don't use antibiotics to treat viruses. So bacteria are alive, viruses are not. Bacteria are free living cells, viruses are non-living collections of molecules. That's how we say non-living collection of molecules. Bacteria are larger viruses are submicroscopic. This might not be quite accurate because there are viruses that can reach the size of bacteria, but still they do not have a cell. So they're not really like virus. They're not cells. Bacteria are self-sufficient, which means they can live like outside, except of course chlamydia and some mycoplasmas that need to be inside of the host cell. Viruses technically, even though they can survive, they are considered like they can't be alone. They can't live alone enough. Like they need to have host cell because they need host machinery. Bacteria have to be like in bacterial infection can be prevented with antibiotics. the best way to prevent viral replication is vaccine, which unfortunately these days people don't get. And just a few more notes on bacteriophages, simple because I like them and I think they're very interesting. So that lysogenic cycle will be asked. So what is phage? So phage or bacteriophage, it's bacterial virus. So it affects and replicates inside of the bacteria and impacts bacteria as a host cell. Bacteriophage enzymes destroy bacterial cell wall from outside, and bacteriophage T4 is one of the 7 bacteriophages. So T1 to T7 are famous E. coli phages, which were found in long time ago, like in 1994. And T7 phage is a famous bacteriophage that I believe looks similar, but this is the famous picture that you will see everywhere. One thing that characterizes bacteriophages is extensive gene exchange, and there is the absence of homology as well between phages because it's a set of genes like explained. So the question is, those are structural similarities in phages because there are many structural similarities. So they literally in phages there are very small variation between virion structures. So are those structural similarities in phages result of something called convergent or divergent evolution? So this is just an evolutionary question, which again comes to the how they evolve, how bacteriophages evolve. So they're all similar, but is that result of divergent or convergent evolution? So what means divergent evolution? That means that all viruses share common ancestor, so they're all similar, right? And that means after they evolve from common ancestor, they maintain basic architecture of some basic structural proteins, which is the reason why they are all literally the same. And then they diverge like beyond the level of homology, but structurally they are similar. Or viruses actually do not, and what means convergent evolution? That means they do not have common ancestors. So it's like opposite than divergent. They simply convert toward the structure that was optimal to build infection. So in summary, is it present common ancestor or there was no common ancestor? That would be a big question. So both types of evolution, divergent or convergent, lead to the simple common trait that helps viruses to survive specific environment, which is a host cell. And based on the accumulation of similar structural characteristics, people say that it's most likely to adopt a divergent evolution hypothesis, which means they all share common ancestor. So the common ancestor, and then they all initially share the same characteristic, and then they diverged depending on the environment where they develop. Again, I will never ask what is actually accurate. So because if something is debatable, I cannot really ask you to tell me what is accurate. So again, many evidences say it is divergent evolution. So there will never be a question which one is accurate, but there might be a question, what are evidences of divergent evolution? So similarities between domains. So some phage like PRD1, which is a very weird name. So its major capacity folds similarly to the mammalian endoviruses, so similar protein folding. The relationship exists between double-stranded DNA phages and archaeovirus, similarities of phages with eukaryotic viruses. So even though they are so different, there are some proteins like 5, 6, and 5, 8 that are similar to eukaryotic viruses. So literally what that means, you see similarities in homology between phages and eukaryotic viruses. So that goes in favor of divergent evolution, which means they must have common ancestor. Whether they do or not, I don't know. And I will never ask you to know. And this is the paper that I will post online. It's first from last year. So last year is when I decided to include it in lecture. So it was the importance of viruses for evolution in soil. So viralizing that alleviate microbial nutrient limitation in the soil. Again, the paper is really interesting, so I recommend you to read. So that's called something. This paper, same like Asgard here that we talked about last class. So this is how we actually know like how we establish the importance of viruses for evolution generally. It's called viral shunt in soil, and this I really asked. I asked last year, and I will ask again. So, what means viral shunt? So, and again, you can read the paper, but I will give you a briefly overview. So, basically, there is like a viral lysis of soil microbial community. So, you have viruses that will lyse bacteria in community. When that viral lysis happens, that increase DOM, which is dissolved organic matter in the soil. And then what they did in paper, they analyzed dissolved organic matter in the soil, and the first question they ask is, does viral lysis of bacterial cell affect soil nutrient characteristics? And the answer was yes. So that will happen thanks to the viral shunt in soil. So again, just a different so they did analysis at two different temperatures. When the viral lysis, they removed nutrient limitation, which means that the viral lysis of microbial soil community improved nutrient content of the soil, which means that viral lysis increased dissolved organic matter in soil, which means that survived microbes can reutilize this dissolved organic matter, which results in increased metabolic activity in soil, which is better for soil health. So basically, in summary, the importance of viruses in evolution, like in addition to the viral shunt. So widely accepted RNA hypothesis. So RNA is the only molecule. So again, just why we think viruses are important for evolution. So RNA is the only molecule that can do reverse transcription from DNA, and that can happen in viruses thanks to the enzyme reverse transcriptase. Some RNA viruses whose replication relies on RNA-dependent RNA polymerase. So this RDRP, which is exclusively viral enzyme, is really this is again goes in favor of acceptance of RNA hypothesis of life. And because of this connection between viruses and RNA, and obviously unique characteristic in terms of RNA and viruses, we think that viruses might be a key for the construction of evolutionary history. And also reverse transcriptase, again, the only enzyme that can actually make DNA from RNA that exists exclusively in viruses. So again, usually you get copy of RNA from DNA, but this is doing opposite, which is why it's called reverse transcriptase. So that all gives the idea of RNA hypothesis as the most accepted one, and viruses as important player in shaping the evolution of life. And that would be all from viruses, which concludes your